Thank you very much. Good afternoon, and appreciate the invitation of the organizers to share some of the thoughts today. So let's get started. Infectobesity, or obesity of infectious origin, is the title of my talk today. But let me start by this very famous quote from a wonderful researcher, very respected researcher in the obesity field, Dr. Stunkett, that most obese people will not stay in treatment for obesity. Of those who do stay in treatment, most will not lose weight. And of those who do lose weight, most will regain it. And the sad part is, this quote was written in 1958, and the situation almost 60 years from then, since then, is not too different in broadly speaking. And I can attest to that personally. I had first-hand experience with this when I treated for my first 10,000 cases for obesity, for weight loss, and I became acutely aware of that sentence and the frustration that I felt in having to ability to do so little for such a big problem, and the repeated weight gain and regain and so on. So the way that was about treatment, but the way I describe prevention in my mind is primary, tertiary, secondary, and tertiary prevention. Primary prevention would be don't gain weight, don't become obese in the first place. Secondary prevention would be if you can hold on whatever weight you are and don't gain further, that would be secondary. And the tertiary would be once you lose weight, don't regain. So prevention could be applied in 3 different levels. Now let me mention one thing. I would like to make a distinction between obesity treatment versus research. What I'm referring to is just exactly like treatment for cancer or HIV. We know that we need better treatments than what we have for cancer. We need better treatments for HIV. But that doesn't mean we drive people away home who are suffering from these conditions. We offer them whatever best we got. And then, keeping this in mind, on the other side, we keep research active so we can come up with better and better treatment strategies or prevention strategies. I think same is applicable for obesity. We need to offer at this individual to community level whatever we got today, but keeping in mind what we got today, if it were acting well, we wouldn't be in this situation in the first place. So here is my thought. I like to keep this in my mind, a sentence, that just one big meal does not cause obesity. So focusing on that, now I'm talking about research. And just one small meal does not fix obesity. So if your research is able to come up with a way to one meal reduction in calorie intake, that may not mean anything in terms of preventing obesity. And very quickly, I would like to point out an argument that I often hear when I talk about biology of obesity and various factors that you'll just hear from me in a second about. I often hear a counter argument that, well, it is your hand that holds that fork and that puts that food in your mouth. So can you not, so it's under volitional control, so can you not eat less? So what I submit to you, my response is, can you eat less? Voluntarily? Yes, you can. So is eating under voluntary control? Yes. Volitional control? Yes, to some extent. But so is your breathing. You know, if I say that you are breathing 18 times a minute, typically you would breathe 18 times a minute, let's just say, and I say that you've been breathing too often, start breathing 12 times a minute instead of 18. Can you do that? Is it under volitional control? Yes, it is. The question is, how long will you be able to do that? A minute, 5 minutes? The day after day, month after month, and year after year that you have to eat less to prevent and control obesity or treat obesity, that is what makes this condition, this disease, very difficult to handle. That is what I want to draw your attention to. And therefore, there is this surge, all of us are searching, effective and meaningful treatment approaches. Not only those that are good in theory, good on paper, but they need to work, actually be effective and meaningful in that sense. So I feel that obesity is a complex disease with multifactorial etiology. And if its etiology is multifactorial, then it should require a multifactorial treatment and prevention approach. Think of you, well, let me just get through this slide. I don't want to run ahead in my thoughts. So I submit that if we have cause-specific prevention or treatment approach, we may get better results for prevention or treatment of obesity than we are getting. And let me explain to you what I mean by that. Here's a paper that describes, in addition to eating excessive or less exercise, there are 10 other putative contributors to obesity epidemic, and there is a list of those right there, including infections, endocrine disruptors, and sleep debt, and so on. Some of those are preventable, some of those are treatable, and some of those are not. But if we have such putative, all right, but if we have so many contributors to obesity, think about our treatment today is blanket treatment. Eat less, move more. In some cases, drugs, and rare cases, surgery. This is almost always regardless of the cause that may be contributing to that obesity in the first place. So if we do have cause-specific treatment, we may get better results. That is the thinking behind this particular talk that I'm going to present today with an example. For example, I'm going to use this putative contributor, infectious diseases. So the question, is obesity an infectious disease? Similar to a question that was asked a while ago, is gastric ulcer an infectious disease? And in fact, I grew up in India, went to medical school in India, where I learned that causes of gastric ulcer are hurry, worry, and curry. And it was very easy to remember the spicy food and anxiety and so on and so forth. But now we know too well the contribution of Helicobacter pylori, thanks to these two gentlemen and who won the Nobel Prize for their discovery of contribution of Helicobacter to gastric ulcer in 2005. So I want to talk a little bit about infectobesity, or obesity of infectious origin. And the significance of this particular research is that, as I mentioned, obesity has multifactorial etiology, but treatment is not cause-specific. So if we do indeed have, if we identify infectious cause of obesity, the preventive part would be much simpler than what we are struggling to achieve today. Think of smallpox. Eradication of smallpox did not require too much of behavioral change, but a vaccine. So I'm referring to that particular segment of population that could be prevented from obesity. The story about infection and obesity goes back to Bombay, India, and it's too long a story for today, but let me just say that serendipity led me and my colleagues to identifying an avian adenovirus, which is termed SMAM1, S-M-A-M-1, that causes obesity in chicken model and also is associated with human obesity. Fast forward number of years, and in the United States, we were able to identify a human adenovirus called AD36, or adenovirus 36. And that causes obesity in animal models and shows association with human obesity. And I'm going to talk about that. But I do want to mention that those are not the only two pathogens or microbes that have been shown to be associated with obesity. There is a long list of other pathogens that include viruses, that include prions, and bacteria, and so on, in various models have been shown to cause obesity or be associated with obesity. Now, this is slide specific to the human adenovirus 36 or AD36 or 8036. These are just some of the animal experiments we have done, and the blue bars show the obesity induced by these viruses compared to the yellow bar that is the control, uninfected control group. And just as an example, here is a study that we did with marmosets, small monkey. They are non-human primates, weigh about 300 grams. And you will see the red bar is the weight gain in infected group. That's the red bar up there, red line, compared to the blue line that is uninfected group, how the weight changed after 8036 infection and the increased body fat gain as well, just as an example. What's the mechanism of action of virus-induced obesity? We and others have done research over the years, and there have been many publications. And if I were to summarize, this is a story about 8036. It appears that this virus infects your stem cells or adipose tissue-derived stem cell and turns them into adipogenic lineage, commissions them towards becoming fat cells, and promotes their number, increases their number, and promotes fat accumulation in these fat cells. That seems to be the overall underlying mechanism how this obesity develops by this 8036 virus in animals. Now, the real question is, does this 8036 cause obesity in humans? Now, that's the million-dollar question, because it is very hard to determine causes of obesity in human, because it has, first of all, insidious onset, multiple etiological factors, and of course, most importantly, ethical reasons preclude any experimental infection. So we had to go by indirect evidence and determine association. Now, here is a twin study that we conducted. David Allison, he was at Columbia University, sent us serum samples of twins, and we started screening them for exposure to 8036, for looking for antibodies. And those that were twins, that both were positive or both were negative, concordant twins, we set them aside and focused only on discordant twin pair. So one was exposed to 8036 and other is not. We found 20 monozygotic and 7 dizygotic twin pairs, and turns out that those twins who were exposed to 8036 were fatter compared to their own twin pair that was not exposed to 8036. I think that is the closest one can come to determining role of virus in humans, short of infecting them. But there have been a number of cross-sectional studies, including this longitudinal study, where it shows a follow-up of 1,400 people for 10 years, showed that people who were exposed to 8036 at baseline had significantly greater fat percent, and they gained in 10 years more fat as well. At this point, when this paper was published, there were about 12 studies about the role of 8036 and human obesity. 10 of them found an association, 2 of them did not. There have been more studies since then, I think close to 20, reported from all the continents except for Africa by various research groups. And there has been a meta-analysis done that concluded that 8036 is associated with increased risk of obesity in humans. Another meta-analysis done by another research group concluded the same thing. So the implications of this is, first of all, obesity prevention and treatment. Here is a study from a Korean researcher. They identified that MCP1 is a marker of inflammatory cytokine, which is responsible for inducing obesity. And their study showed that if you have mice that don't have MCP1, that is no inflammation, they won't be 8036 induced obesity. So taking it from there, they used another experiment. They used an anti-inflammatory agent, mulberry extract, to show that mulberry extract can treat AD36 induced obesity in animals. And recently, they went on to show that if they develop a vaccine by inactivating a virus and then challenge those animals with that virus, they can actually prevent AD36 induced obesity in animals. That is their proof of concept study. So I like to submit to you in conclusion that I like the word obesities because there are so many different types of obesity that may exist. This suggestion has been made in the past. And I like to say that because here is one example, as I said, unique prevention and treatment strategies could be used for AD36 induced obesity. And remember, although there are multiple microbes that may be responsible for obesity, we only talked about one. Who knows how many may be out there and what segment of population may be affected by that contribution. And so, therefore, I come back to this, that if we have cause-specific treatment, and to have that, we need to identify what are the causes in the first place. Anyway, here I conclude my talk. I would like to declare a potential conflict of interest. We have number of patents filed. If anybody is interested, I can give the details later. We have received grant funding from number of sources, commercial as well as not-for-profit organizations. And my collaborators, current collaborators on AD36 induced that area of research, and numerous collaborators and contributors that I'm grateful to over the years that we have produced these studies, and these are people from my lab last year. And thank you very much. Much work to do. That is what I'm pointing out here. And if there are any questions, I would love to.
Z-Hawk with
yes.
Z-Hawk with Mississippi State University. Very, very interesting presentation, I must say. The relationship between AD36. Can you hear it?
No, I can't see you.
Oh, I'm here. I'm right here. I'm a backbencher.
Oh, yes. Right.
Yeah. So the relationship between AD36 and obesity, you said that you've looked at MCP1 and relationship between MCP1 and AD36. Has anyone looked at AMPK and relationship to AD36? Because AMPK is in the front line nowadays. As you know, people talk about AMPK and obesity as it relates to lysis and genesis of fats. Would you shed some light on that, please?
Yes, AMPK as say in the liver, so MCP1 as inflammatory cytokine. That study was done by Korean researchers. We had nothing to do with it. But we have done AMPK, we have looked at AMPK in our animals, and phospho-AMPK in the liver is significantly upregulated in presence of AD36 infection. That's a completely different topic, actually, because AD36 also improves glycemic control in certain situations. But I didn't want to, that's a very different topic. And in that connection, that phospho-AMPK appears to be acting.
I wonder if there's any longitudinal epidemiological data showing an increase in infection rates with AD36 that might correlate with the onset of the obesity epidemic.
Yeah, right. That would be a systematic study done over the years, and that is, in the United States, that's lacking. There is a study published from Sweden, but they follow different populations, blood collected in different cohorts, and it shows increase over the last 50 years. But there were different populations collected. And I'm not aware of US data that shows, okay, it was 70, 80, 90, 90, how it has changed over over the years.
And also, at any recent time points, do you know what the, from immunological data, what percentage of Americans have been exposed to 8036?
Yes, we know that. We have done multiple studies and others have done studies. If you put all of them together, it comes to about, cross-sectionally, it comes to about 17% of the US adults are infected with obesity. There is a little higher percentage in children of exposure to 8036. And if you break that down into obese versus non-obese, we had one study done that way, where we saw, so collectively it's about 16 to 17%, 30% in obese, and 11% in non-obese. So if you break them down by obesity, it is different, more in obese, less in non-obese. But the interesting part was, in obese individuals, those 30% who were exposed, they were fatter compared to the unexposed among the obese. Among the non-obese, although 11% were exposed to 8036, they were fatter compared to 89% unexposed as well. So even within that non-obese category, they were heavier compared to their unexposed counterpart.
I'm wondering if your animal studies tell you anything about windows of susceptibility, whether young animals are more susceptible.
Excellent question. Yes, indeed. Younger animals are, their response is more dramatic and they are more susceptible. And I think that has to do with the mechanism that the virus seems to be turning on these resident stem cells in adipose tissue and making them fat cells and multiply them. I think that has something to do with that age in particular.
Could you do me a favor and comment on the onset of infection and the time course of obesity possibilities in terms of time course, how long obesity onset develops?
It is, I would rather give it to you using animal studies. In animal studies, it is, every animal species is different, responding differently. If you talk about chickens, it is 3 weeks. If it is mice, it is 1 to 2 months. In marmosets, it is like 1 or 2 months. But the most interesting was rhesus monkey study that we did when I was at University of Wisconsin Medical School, where we had natural exposure of rhesus monkeys to 8036, natural exposure. We didn't infect them. And it turns out these adult monkeys, once they were exposed to 8036, within 6 months, they gained 15% of their body weight. So it was that quick. All right, thank you.